Okay,
Yes,
then I can start.
yes, please.
Yeah, so hello everyone. My name is Gaurav Chaudhary. Um, I'm working at EPT, Energy and Process Engineering in Gløshaugen, in Trondheim, and I'm in this group called Enersense. Um, just a small intro to myself. I come from New Delhi, India. I did my bachelor's in IIT Roorkee. I did my bachelor's in architecture. Then at NTNU, I came in 2017. I did my MS in Sustainable Architecture. And then since January 2020, 2020, I'm PhD. So the topic of my PhD is data-driven optimal control for building energy systems. So the main objective is to push the state of art in design and implementation of real-time data-driven predictive controllers. Predictive control is a different model-based predictive control is used in chemical engineering. A lot of automation uses that. Tesla auto driving uses that. When I say is to push the state of the art, so work is already there. It started like 5 years ago in this field in data-driven controllers, but it still needs a lot of work. So my main objective is to solve the problems which people face when they actually implement real-time controllers in buildings. So till now, most of the people are only in research. So, but the good use is to how can we implement in the field. Um, for this course, I want to do this um, project called training data relevance in sequence to sequence based LSTM models for indoor air prediction. So, my main objective is to develop a multi input, multi output sequence to sequence LSTM model based on encoder decoder networks. So, I want to take the data of past 24 hours and predict next 24 hours. So, why I'm using this? Because, as I said in the beginning, I want to create this for predictive controllers. Predictive controllers needs data for next one day or half a day, so we need to be able to predict future at least one day. So I'll start with one day and see how the accuracies go. Maybe I'll reduce it to half a day or 18 hours, something like that. So the first objective is to develop it, of course. The second objective is to study the data, study the model prediction accuracies when you use different temporal quality in the data set. So the data which we get from the buildings, indoor data, occupancy data, heat flux data, amount of solar radiation falling in the window outside. So all of that is comes in different temporal quality. Temporal quality means time step 10, 15, 30, 60 minutes. So I want to study how does the model prediction accuracies change with different time steps. Why this is useful? So that when I actually implement a real-time controller, the real-time controller will have a possibility to capture the data from the inside of the building. So with this kind of study, I'll be able to guess or make a good assumption. How often should I record the data and how often should I average the data? Another possible what I want to study in this study is, okay, once I sell this real-time controller to an occupant in a house, when should he start training the model? Should he start training that model in January, in February, in June, in October? When should he start training the data, training the model? And how does accuracy change for the subsequent months? So if I start training the data, training the model in January, how does it change in the subsequent months? So why this is useful? So that you can find an optimal time when I can sell. Because if I sell the real-time controller to a house, for sure he won't be having the indoor data of his house for past months. So he has to record some data before the controller becomes self-aware. Self-aware is a big term, but kind of becomes better. Uh, for now, I'll be using the data set which was available in 2020 on Nature dataset. It's recorded in Singapore by office building. It has the whole data of 2 3 years. I don't exactly remember, but for 2 years it's very good. And it has a lot of data which I can use. It's because it's real building, it's better. Of course, I can always generate data with many simulation programs, but I want to use. But then I also again fall in the same realm that I'm working with the research data, working for the research, so nothing is implementable. So I want to use real data from real buildings. I had already submitted this abstract. You can see it. I submitted in for this conference called CISBAT 2021. It's Carbon Neutral Cities, Energy Efficiency and Renewables in Digital Era. It will be in September 2021 in Luzon. I submitted in 24th. I'm waiting for the abstract acceptance. I hope I'll get it. I think I'll get it. And then by 30th of April, I'll be submitting the paper. So it falls really in the timeline of the whole course also. Yeah, that's it. Thank you.
This is very good. Thank you very much for the nice presentation and the nice task.
Thank you.
So any questions for? Please unmute yourself and just if you have any questions, you can ask it. So I have a question, in fact, about the data set. You said you have 4 years data, right?
It's around
3 years.
it's around 3 years. Yeah, I can try to show you if my Google.
So of course, you need to start by plotting the data, how it looks like.
Yeah. I can try to show you. So it says it is from July 2018 to December 31, 2019. So
Okay.
18 months. Okay.
And
it has, so it comes in 1 minute interval. So I can do that study, which I said, studying different intervals. So I can average data for 10 minutes, 15 minutes, 1 hour.
And it's from Thailand, Bangkok.
Yeah.
So is it possible to use this data set here in Norway, for example?
Yeah. The thing is, so I thought about this, that it's different climate. Right now, my objective is to make the model work for any climate and do that study for any things. Then I'm in contact with Skanska and some other companies in Norway, and I'm trying to get building data set for their buildings. So if I'm able to get that, then of course I can work on that also. But right now, I only have at least not non-Nordic data. Yeah.
Okay. So I want to inform you something. You know, scientific data, they publish the data set itself.
So this is scientific data.
Yes. So when you, and it's publication and it's an open access, and I'm sure that this paper will be highly cited. I mean, it's just in 2020.
Yeah.
So, but already more than 1,500 persons, they got access to this article.
Yeah.
So when you collect your data, you need to publish your data set as well. So other people can use it in Norway and outside Norway, for example, and it be counted as one publication.
Yeah.
So the data itself is value, and if you can manage to publish this data set similar to this publication here, it will be very, very useful, in fact.
Yeah, so my plan is to publish that paper in the conference for sure.
No, I'm not talking about the paper, I'm talking about the data set.
Because the data set is already published, so I cannot republish it, right?
No, I mean the data set which you will collect from Norway, for example.
Oh yeah, from the Norway, yes. That is, yeah, I'm part of a IEA annex. IEA is International Energy Agency, and there are different annexes for that. So yeah, I plan to publish it there. So something like this.
This is very good. This is very good. And I want to ask also, is there any difference number of people who are living inside? So if I'm living in my home with my family,
so when I say that I'll be collecting the indoor data also is the occupancy data. So because occupancy changes the way indoor building physics works, so it is a big factor. One human being is considered like a 120 watt bulb, which produces energy. So that is, of course, always a part of the
Mhm.
factor.
Because I have been working in greenhouse climate control, and
Yeah,
we have planets inside the greenhouse, and they grow up. So each time, because they produce energy as well. So we need to consider this also. We need to take this into account
yeah,
by measuring inside temperature and.
yeah,
Well, this is very nice task, and I guess it's you can manage if you need to submit the full paper in March or in April.
yeah.
So it should be possible to. So maybe the next what will be the next step for you?
Sorry.
What is the next step for you?
So I'm almost done with this. Um, as you said in one of the slides, under overfitting the data. So I was facing some problems in overfitting here, but I managed to solve with L1, L2, and some dropout.
Mhm.
Yeah, I still have to work a bit. I think I was taking testing data a lot less than validation data. I was just trying to see how model works with less testing data.
Yeah.
Sorry, training data.
Mhm.
And when there was very less training data and a lot of testing data, the it became underfit,
But
something like that.
it's time series, right? So
Yeah, it's time series. Yes.
you need to be careful.
Yeah.
So you cannot go for. I mean, you cannot shuffle the data. You need to be careful.
Yeah.
Just need to keep the sequence. You don't need to destroy the sequence of the time series.
Yeah.
Mhm.
So once I'm done with this, then I can make different data sets for averaging and all this study.
Mhm.
Yeah,
So I guess you can start writing, working in your paper now, and the
yeah,
result.
exactly. I will actually start in a
Yes.
week or this week for
Mm.
sure.
I mean, it's a conference paper and it's short, but this paper should be extended into a journal.
Yeah, it can be, but I was thinking that this kind of work, where the main programming work,
Mhm.
this has been done by not done a lot by a lot of people in my field, but in general field. So this would not be a big kind of a research objective or something which I'm new to the world. So
No,
the
no,
new which I'm considering is this part.
but it's the readers of the conference. So I mean, it's not machine learning conference. Maybe if you
Yeah.
are submitting it to a machine learning venue,
Okay. Yeah.
for
Then it
example,
makes
maybe it will
sense.
be rejected because it's not state of art. But
Yeah.
in this area, in this domain, maybe it's state of art.
Yeah. Yeah.
So,
It is
so
for sure.
it's according to the audience who will read my paper. So,
Exactly.
for example, in agriculture, if you can just use some prediction or some classification of leaves and stuff like this, it will be just fantastic in this area, and they will just accept it, and they will just say, "Wow, this is very good," because
Yeah.
it's a new application in this domain. But in
Yeah.
itself, there is no any innovation because. But we
Yeah.
try to use models from different areas and try to use it in your domain. So maybe in your domain, I mean, as a journal paper, maybe you can just go for more models and make good comparisons between different types of models.
Yeah.
Maybe you can add the control part, the predictive controller, and
Yeah.
Mhm.
Yeah, so that is my so for my PhD, I plan to publish 3 journal papers because
Mhm.
3 is the max minimum you have to have.
Mhm.
So I'm not focusing a lot on writing a lot of papers. I want some good work and just write 3 journal papers. So in the future, my target is to make a second paper. The first is going to be my review paper.
Mhm.
The second will be control based on this model.
Mhm.
So that will be the journal papers. But this I just want to publish as a conference paper and get some ideas from people there and
Mhm.
ideas from, for example, you and other
Mhm.
people here.
Yes.
Yeah.
Okay. So thank you very much.
Okay, thank you.
Yeah. Thank you.
I can stop the screen, I guess.
Yes, please.
Yeah.
Who next one to present today? We have maybe 10, 15 minutes. Who next? Who wants?
I could try to present if I can
Okay.
keep it brief since I have a meeting at 12.
Okay. So I
Yeah.
will make you as a co-host.
Yes.
Mhm.
Good. Okay. So, first of all, I can start with a brief introduction of myself. My name is Irini. I am coming from Greece. I'm affiliated in the Department of Biology in Trondheim, but I'm currently working from France. I'm working remotely. And I think, more or less 1.5 year into my PhD in computational biology. I'm working with the development of models that represent different cell types, and these models can be used to perform different simulations and acquire predictions for the behavior of the cells under different perturbations or different treatments with drugs. So it's a way to aid experimental design and also be able to test multiple hypotheses in a more cost and time efficient way. So as part of my PhD, I'm working with the development of models for psoriasis, chronic skin inflammatory disease. And based on that, I have, in general, psoriasis is a really heterogeneous disease. It has really variable manifestations in the clinic. It has different severity grades, and also many patients respond differently to treatments. This is the case, of course, for many diseases, including cancer, and a way to solve this problem of differential response to treatments is the molecular characterization of a disease and the classification of patients into different subtypes. Based on this classification, we can use them to personalize the treatment plans for different patients and choose these treatments that are more probable to work best and have lower side effects and so on. Currently, psoriasis patients are only grouped together based on the severity and what's the area of the body that it is affected by the disease. So my idea was to use gene expression data to classify patients. I was thinking that there might be some genes that can distinguish different subgroups of patients, and these genes might be correlated, these genes or features can be correlated as well with, for example, the onset of the disease, what is the age of a patient will start developing these plaques on the skin, if these subgroups or subtypes of the disease overlap with treatment response. So we know, for example, that if a gene is produced more in a patient, then this patient will not respond to some specific drugs and so on. So I looked in computational biology, we have many repositories of publicly available data. So I collected a few examples of data that are biopsies, meaning they have taken a piece of skin from volunteers. Some of them have psoriasis, some of them are healthy. And so usually the biopsies include either psoriatic samples that they take a piece of the skin where the plaques of psoriasis are developed. They also take adjacent tissues, so a piece of the skin from psoriasis patients, but that does not have the characteristic plaques for the disease, and healthy donors that could be anyone. So my idea was to maybe this is too ambitious. It was something like I came up yesterday. So my idea was to take this gene expression matrix. So imagine a data set where you have different patients as columns and different genes and their expression as rows. And we are talking for quite big data sets. We have 21,000 genes that are expressed and are usually measured. So as a first step, I thought of do a dimensionality reduction, maybe choose the most variable genes, for example, take the 500 top genes that are most variable across patients. I'm not sure about this step exactly. And based on that, I thought of doing a clustering, maybe k-means, in order to acquire different clusters of patients. It was already mentioned today that choosing the right number of clusters depends of expert knowledge, but I have read similar publications where the number of the cluster is defined as such when the all healthy donors are uniquely in a single cluster and no other samples are in the cluster of healthy donors. So that was an idea and something that I could try to apply. So it's kind of a heuristic process here to find the number of clusters. And using these clusters, I could identify the futures, meaning these genes that distinguish those clusters, use part of them as the majority of those as training data for a random forest classifier. And of course, part of them could also be split for validation data, and then use the other public data in order to assess the performance. So for target journal, I didn't have something specific in mind, so any bioinformatics or computational biology journal would be fine. And about the timeline, I also do not have one because yeah, I'm not sure how long each steps would take and whether this also something too ambitious to aim to complete by the end of the course. And that's all for me.
Thank you very much, Irini. I mean, the talk is very, very nice, and it's very, very important because it's our safety, and so this is very nice. So you decided to use K-means clustering, so it's unsupervised, and we random forest or any other models.
Yeah.
So and the graph which you presented is very nice. I like this way for presenting things because it make it when it's
Thank you.
visual. In fact, it make life much easier.
Thank you.
So I propose it to everyone to try to visualize his model. So when he try to solve a problem, he try to develop similar graph, for example, to representing the whole process from A to Z. Um, I have a problem. I mean, how many healthy gene expression? How many has developed the disease? Because I feel that maybe we will have this unbalanced data set.
So I have identified a specific data set that has 92 psoriatic patients and 82 healthy.
Yeah.
But
And
indeed,
10 was developed as a disease, right?
sorry?
It's 92 patients,
Yes.
82 are healthy, and the 10 they
No,
developed.
no, it's 92 with the disease and 82 without the disease. So it's
Okay, okay,
174, I think. Yeah,
okay.
yeah,
This is very good. No, no, this
yeah.
is okay. So sorry for my question because I
No problem.
understood that it's 92 in total, 82 are healthy, and the 10 are developing.
No. It's yeah.
No, I mean the project is fantastic, and I mean the
Thank you so
journals which
much.
you selected are very, very good. And
Thank
I
you.
guess it should be possible by the end of the course, at least getting results and.
Good to hear. Thank you.
But the gene expression, how it look like?
So gene expression, it's actually, as I said, you have the patients and whether they're patients like with or without the disease as a columns, and then you have a genome-wide gene expression, meaning that we count for the expression of all genes that are expressed are usually 21,500, I think.
Mhm.
And then it's numbers that can vary from 0 to whichever number usually you have around 50 and so on.
Okay, so is it possible to normalize and to apply other methods?
Yes, exactly. Yeah.
Mhm.
So yeah, I think it might be a need for aggregation of these data sets
Mhm.
in a way, and normalization or standardization for in order to be able to combine everything, because they come from maybe different platforms that
Mhm.
are sequenced and so on. So I expect many batch effects,
Mhm.
but it's something, yeah, I will try and see
Also,
how.
total number of samples will be 174. So I guess it's important to go for k-fold cross validation.
Okay, yeah.
because it will be very expensive to put hold out. It would be just smart to go for folded course variation.
Okay, good. Thank you for the suggestion.
Any other questions from any other? Yes, we have some questions. Maybe someone, Muhammad. Yes, Omir. Omir.
Hello. Yeah. Actually, I raised my hand to present.
Okay.
Yeah. But I think the presentation by Irini, yeah, it was very nice,
Mhm.
and I'm also working on some deep learning models. My PhD is on digital twins, and I'm working on 3D reconstruction and model retrieval using some deep learning models. So uh, I think it would be great because there are many people who are currently, I think, working on different deep learning models, but I think we don't have much time to present today.
Yes.
What do you think, Ibrahim?
No, no. I mean, today we need to finish at 12 because some people they have some meetings at 12.
Yeah. Okay.
Yeah.
Okay. Thanks.
So we will continue next Wednesday. But I wish also people to talk to each other so they can help each other in coding, in developing algorithms, testing, and so on. So people can share knowledge and work together and get some co-authorship from each other. It would be very, very nice. So it would be nice, for example, if someone working in biology, working in medicine, and they have someone from engineering, from ICT. So I guess it would be fantastic. So we can support and help in coding the part if it is required. I mean, okay, so thank you very much for today, and we will meet again next Wednesday. It will be the same also. I will have some lectures, and after that we will have some project presentations. But please, I mean, try to use the knowledge which we presented today. Try to visualize your data. Try to play with principal component analysis, and step by step we will go into deeply into those those topics concepts. Yes. So thank you very much for today, and we will meet
Thank you so
next
much.
Wednesday. Thank you, Irina. Thank you. Thank you for everyone. Bye bye.